So we're going to pick up with photosynthesis, and we're going to start here. And so what I showed you guys last time was really half of the light reactions. And what I want you to do is help me identify all the parts of the photosystem and then how it is they're used in connection with the rest of the light reactions. And understanding photosystem 2, which is what this photosystem actually is, will help us with the rest of the light reactions. So this is the photosystem, right? So we have some protein matrix into which chlorophylls are embedded. And remember, chlorophyll is just another protein. It's a really large protein that has a magnesium ion associated with it. And that magnesium ion is actually what picks up energy from photons. Okay, so here's our photosystem. This black outline is the protein matrix into which these green things are embedded. Those green things are chlorophylls, right? This is P680. And please remember, it's an incredibly strong oxidizing agent, which means that it really wants to take electrons from other things. And ultimately what's going to happen is it's going to take an electron from water. After something happens, I'm actually going to erase this a little bit. And then there was this large protein complex up at the top of this photosystem. And what's that called?
Primary electron acceptor.
The primary electron acceptor, exactly right. Which is an incredibly descriptive name. It just accepts electrons. That's what it does. And it passes them to the next stage in the light reactions, which is an electron transport chain. Now I gave you last time a function of all electron transport chains. And it doesn't matter where that electron transport chain is located, whether in photosynthesis or cellular respiration, they all do exactly the same thing. And what is the function of an electron transport chain?
To pump protons to create a proton gradient.
It's just making a proton gradient. It's going to take protons from one side of the membrane and pump it to the other side of the membrane. That's all it does. But that proton gradient is really important because it becomes a form of potential energy. And that potential energy is used to create some incredibly important compounds. Okay, so we know that chlorophyll is sensitive to photons, and the sensitivity ranges, right, depending on the type of chlorophyll we're talking about. So chlorophyll A has a slightly different absorbance frequency than chlorophyll B does, but generally speaking, they're sensitive in the blues and purples, the blues and violets, and then down kind of in the orange and reds. And it's not terribly sensitive to either the greens or the yellows, right? Okay, so a photon is one of those important wavelengths, either really, really short or really, really long, comes along, and it, we're going to call this a photon, and it strikes a chlorophyll molecule. What happens to that chlorophyll molecule? What does that mean, that it gets excited?
It has some of that, I don't have to say energy, but that's not exactly right. It takes some of that energy and it bounces it between the rest of it.
Exactly right. So some energy is going to be passed between chlorophyll molecules, but that happens because something changes molecularly to the chlorophyll molecule. And what actually changes? And this is how we define excitation. Something moves. What moves? An electron, exactly right, on that magnesium ion, right? One electron does what? It moves out one energy shell, exactly right. So that's excitation. A photon that's carrying some amount of energy strikes that magnesium ion on the chlorophyll molecule, and it causes an electron to move out one energy shell. Remember, that represents energy, because the farther an electron is from the nucleus, the more energy it represents. So an electron that was at some lower energy level absorbs some energy from that photon, and it moves out one electron shell, right? That's excitation. Now that energy is released when that electron falls back to its ground state, which is that original electron shell. And that energy is picked up, and please remember, it's just energy. That energy is picked up by adjacent chlorophyll molecules. So at this point, we just have energy passing between chlorophyll molecules, and ultimately, it passes to what's called the reaction center core, which is this middle part of the photosystem. So that energy passes over to this molecule, P680. Now P680 is just two large chlorophyll molecules that are rammed together. And they have this unique absorbance spectrum right around 680, on average, 680 nanometers. Now this is going to be unimportant, but I need you to understand why that 680 idea is a thing. So if you think about the kinds of wavelengths the chlorophyll is sensitive to, it's really sensitive to super low wavelengths, right? Like 460 nanometers, which is way below this average. It's also somewhat sensitive to longer wavelengths. But you guys, every time energy is absorbed and converted and released, some energy is lost as heat. So what that means with respect to photons is the wavelength becomes longer and longer. So by the time it gets down here, that photon's energy is really a correlate of a much longer wavelength, because some of that high energy stuff has been lost through transferal of energy between chlorophyll molecules. Now I won't ask you to regurgitate that on the exam, but I want you to understand why that P680, that 680 nanometers is a thing. Okay, so P680 receives that energy, it becomes super excited, and it becomes so excited that what happens to it? It loses an electron. Is that a reduction or an oxidation reaction? It's oxidation, exactly right. So at this point, P680 is oxidized. Now, P680, super unhappy when it loses electrons, and it's an incredibly strong oxidizing agent. So what it does is it takes a water molecule and it splits that water molecule. It takes an electron off of the hydrogen in the water molecule. And if you take an electron away from a hydrogen, what is left? Remember, hydrogen has the atomic number of 1 and the atomic mass of 1.
So just that.
So without any sort of manipulation, how many protons does hydrogen have? Just 1. And if it doesn't carry a charge, how many electrons does it have? Also 1. And if it has an atomic mass of 1, how many neutrons does it have? Close to 1. 0, yeah, because it's just 1. If it had an atomic mass of 2, which is protons plus neutrons, it would have 1 of each, right? But mass is just the sum of protons and neutrons. So if it has an atomic mass of 1, it can only have 1 proton. So if we take that 1 electron away from it, what's left? 1 proton. So this thing right here, this H+, this hydrogen ion, is really just a proton. That's all it is. That accumulation of protons is really important, right? Because we need to make a proton gradient, and this becomes a source of those protons. Okay, so when P680 tears a water molecule apart and it removes an electron from it, is P680 reduced or oxidized? Close to oxidized. It's reduced, yeah, because it's taking an electron. Water is oxidized for sure, but P680 is reduced. Okay, yeah, Kira.
So, I just want to make sure I'm thinking through this right. So, as the energy goes into P680, is the energy that's going into it what's breaking up the H2O? To get that electron up there?
The energy that's going into it is what's causing that electron to be released.
Okay.
It's just an inherent characteristic that it is an oxidizing agent. It doesn't have much to do with that energy. When
Okay.
it loses an electron, it just yanks electrons away from other things.
So,
It's strongly electronegative is the way it's used.
so is that electron coming from the H2O being
It
burned?
is,
Okay.
yep.
But you need the energy coming from the photon to do that.
You do, because that's what causes the electron to actually be released from P680.
Okay.
So this is photosystem 2. We're going to talk about photosystem 1 in just a second, and I know we're doing it in reverse order, and it's stupid, but it's just how it works.
So this is ATP? Like, is that what you mean?
We're going to eventually make some ATP.
Oh,
so-
that's what's making ATP, and then the photosystem one would be the NADPH?
Correct, yeah, that's what's going to make some NADPH photosystem. Okay, so, oh, go ahead.
This is
So the next thing in the chain of events is our electron transport chain, right? And the way that I do this before was as kind of like a cascade of rectangular proteins. The electron transport chain. Now, these proteins are also embedded in the chlorophyll. Like this. Okay, so the primary electron acceptor has an electron, and it has that electron from P680. Right, so it picked up an electron, was the primary electron acceptor reduced or oxidized?
It was oxidized.
It was close to oxidized. It was reduced because it picked up an electron, right? So it then passes that electron over to the electron transport chain. So when it passes its electron, the primary electron acceptor becomes oxidized or reduced. It becomes oxidized and it reduces this first protein, right, in the electron transport chain. And then that electron is just passed down the electron transport chain until eventually it falls out and weights down here. Now, we're passing that electron down the electron transport chain because that electron actually represents some energy as well, right? It's like electricity. That's all the stuff that comes out of our sockets is. It's just bouncing electrons. So that electron is energizing these proteins to pump protons from one side of the membrane to the other. And these protons, again, have largely come from over here, the oxidation of water. So we get protons being pumped out. Now, one thing that I would like you to remember is these proteins in the electron transport chain are called cytochrome proteins.
Inside the ETC?
Uh-huh. I'll write that right here. Cytochrome proteins. Now that's an important observation because regardless of the organism that you're looking at, whether it's a plant or an animal or a protist or a bacterium, if it has an electron transport chain, it is a cytochrome protein, strongly suggesting some common ancestry between all organic life. Okay, so we're getting a strong concentration of protons on this side of the membrane and a very low concentration of protons on this side of the membrane, which is setting up a diffusion gradient, right? Now, at the end of this chain of events, we have what Max was referring to, a large protein that makes ATP. And that large protein is called ATP synthase. Now, on the exam, I asked you 2 items about facilitated diffusion, or at least that included facilitated diffusion. And remember, facilitated diffusion is just a passive movement because it's diffusion. It's a passive movement of some substance from a high concentration to a low concentration. But it's facilitated by some protein channel or carrier molecule. And that's what's That's what ATP synthase is. It's just a carrier molecule or a channel that allows protons from one side of the membrane back to the other to follow their diffusion gradient. So question, if this is diffusion and there was a higher concentration of protons here than here, would they flow back out of the chloroplast? What do you think?
Can you repeat it?
You betcha. If this truly is just a passive process, if it's just diffusion, and ATP synthase is just a channel that's allowing protons to follow their concentration gradient, if there was a higher concentration of protons on this side of the membrane, would they flow back out?
Yeah.
Yeah, for sure they would. It's just following a concentration gradient. If for whatever reason there's a higher concentration here than here, then they're going to go back out the other way. Generally that doesn't happen, right? Because you have these pumps right here that are actively transporting protons back out. But if some malfunction happens here, you get a higher concentration of protons in here, they're going to flow back out the wrong way. Okay,
Kind of like a fail-safe?
what's that?
Is that sort of like a fail-safe, so if the ETC stops working, it'll almost hydrogen out, as soon as that is back into higher concentration, they'll flow back through?
I don't know that it's a fail-safe, because that would certainly be counterproductive for the chloroplast. And if that's happening, that plant cell's on its road to death. And it's just going to die. Okay, so ATP synthase allows protons to fall back through following their concentration gradient. Like that. So you're going to get protons falling back through.
Is that called the concentration gradient?
Yeah, up here is where we're creating a concentration gradient, and they're just following that gradient from high concentration to low. Now when that happens, ATP synthase physically spins in space, right? So it's a form of kinetic energy. And that kinetic energy is used to take an ADP, adenosine diphosphate, or an ATP molecule that doesn't have that third